Hello. Hi, everyone, and welcome to today's course on an introduction to cross-species neuroimaging. I'm really glad you're all joining us today. This is something that the cross-species community has kind of been pushing for a few years to get an OHBM educational course. So I'm really happy you're all here for the first version of it. And what we're going to try to do today is take you through all the steps of what you need to do, all the way from acquisition and data requirements, the unique parameters that you might need to do in scanning, which is with Steve. And then we're going to move into more and more advanced analyses across multiple modalities, from using everything from structural to diffusion to functional MRI. And it should be a lot of fun. So with that, Steve, why don't you take it away?
Thanks, Austin. So my name's Stephen Sawiak. I'm from the University of Cambridge. I'm a preclinical MRI physicist. So to start us off, as Austin says, I'm going to talk about the acquisition of the images and what we have to do differently if you're used to human imaging. So I'm sorry to say that I have no disclosures or conflicts of interest. So the issues in animal imaging come about from the different scales. So animals that we might image come from large mammals. So the largest I've scanned is a sheep, to non-human primates, to rodents. For the physicists in the audience, these pictures aren't to scale. For the large animals, we're probably going to use clinical systems, in which case there's not that much to change in our acquisition parameters compared to humans, as I'll discuss. For the smaller things, we need to use preclinical systems. These will be at a much higher field. And the physics involved, they're the same principles, but the parameters change, and we have to change the way we acquire our images. So to put this in context, it's no surprise that these different sized animals have different sized brains. Here's a human brain, a sheep brain, and a mouse brain shown roughly at the same scale. So we talk about MRI all the time as being a very translational technique, that we're doing the same thing, but in fact there are quite important differences in what's going on with the signals coming from these different sized objects. The interaction of the RF with the coil and the size of the brain is different. The magnetic properties are different, and the most important thing is that the signal we get is different. It's much smaller. There's less stuff giving us signal. So we have to use some different tricks. So while the underlying physics, the principles are the same, the way it works out is different. So if we have a large animal, as I mentioned, we're probably going to put them in a human system like this. It's slightly gratuitous to show you, but they're some of my favorite slides. One of my favorite experiences in Adelaide, putting a sheep in a scanner. Here it's going into a knee coil, going into a large bore human scanner. This is 120 kilos of sheep, so it took 4 of us to get this guy in. There it is, happily resting away. And the images that we get are very similar to human images. So here are T1 weighted and likely T2 weighted images from the sheep, and we can see that it's not a wholly different story from the human case. For human morphometric imaging, mostly we're going to use the T1 differences between tissues to get the contrast in our images. And just to illustrate how that comes about, one of the most common sequences used, MP-RAGE, inversion recovery SPGR, has an inversion preparation. So if we look at the differences in signal, this would be what the signal looks like after the inversion pulse. So white matter has a shorter T1, and so recovers slightly faster. It also has slightly less proton density, about 7/8, compared to grey matter. And if we choose our inversion time carefully, we can get different contrasts between these. So if I have a very short inversion time, you can see that the difference between them is relatively small. As I increase it, I get maximal contrast, which then goes away, and it tails off with a longer time. If I choose the time, for example, over here, where the white matter signal is 0, then that's completely dark in the image, and we won't see the white matter. We still see the grey matter, so it gives us some contrast between them so that we can see the shape of the brain and the anatomy. If I chose it here, there is no contrast between the grey and white matter. This would be a much less useful time to choose. Mostly, with these images in humans, we'll choose the time around here so that we're nulling the signal from grey matter and we have bright white matter. So the image I'm showing you here would be the inversion recovery signal. That's like the NP rage with just the NP part. There's no rage, and this is with a long TR, so the CSF is still bright in this picture. In practice, we would actually go a bit longer so that the signal's recovered a bit, so the signal to noise is higher. And this gives us good images for the human brain because of the difference in T1 and T2. T1 and proton density, sorry. The problem is with small brains, because the signal's much lower, we're going to have to go to higher field. And at higher field, T1 tends to increase, and the contrast between the different tissues is less. So just to show you that in practice, here's the signal I showed you before. At higher field, the signal might look like this, with a longer recovery. So to get the same contrast, we're going to have a longer time. It's going to take us longer, and the contrast, the difference between the tissues, isn't going to be as good. Worse, in small animals, particularly those with a smooth cortex, there is much less grey and white matter intercalation. So you tend to have a smooth sheet of grey matter sitting on the white matter. They're less mixed up together. So nulling one of those tissues doesn't tell you everything about the shape of the brain in the same way that it does for human imaging. In addition, because there's much less white matter, you have partial volume effects. So if you're looking at fine structures, so here, just under the insula, this is a marmoset, the white matter is really small. So relying just on the T1 difference, it's not going to give us a good picture to infer the shape of the brain. If we look at the values of T1 that we get, so here's a marmoset brain, and I'm just going to show you a movie changing the inversion time through that graph as we saw before. Okay? So for very low inversion times, we have no contrast. We see the white matter's nulled, then the grey matter's nulled, the white matter's bright. At the moment where that happened, there is no signal anywhere. So whereas in the human case, where we could make the grey matter attenuated, the white matter would be bright, we get a good image with nice contrast, and we can see everything quite well. In the marmoset brain and small animal brains, because of the higher field, the longer T1s, at the moment where the contrast is getting good, the signal is getting bad, and the image is not very good at all. It's the same story in the rat. So if I play that movie again, just when the contrast gets good, the signal to noise is bad. So these T1 weighted images are not good for small animals. They don't show us the anatomical differences that we want because the signal isn't high enough. So the NP rage approach is not going to work for small animals. Because of this, it's very common to use fast-spin echo or sequences like that. So these images, again from a marmoset, you can get in just a few minutes, and they look really nice. These are relatively long TR. It's just a bit of T2 weighting, and you can see the contrast largely driven by proton density differences through the brain, and you can see good grey and white matter contrast. It looks really nice. The problem is these images take quite a long time to acquire if we want to do them properly. So although these images look nice, the slices are thick. So if we look at them in horizontal and sagittal views, those thick slices mean we haven't got information through the whole brain. If we're going to do computational neuroanatomy, automated analysis, look at differences between individuals, these thick slices mean although they look nice in the plane, structures that change between the slices are just being blurred out, and we can't see them properly at all. What we need is an isotropic resolution so that it's the same in all directions. We don't want to have these thick slices if we want to analyze these structures properly. With fast-spin echo, this will take us a long time. So the first images I acquired like this is a mouse brain. This is a fast-spin echo sequence from 4.7 Tesla. It has 100 microns isotropic resolution, but that takes an hour and a half. So it's quite a slow, long image to do. We can do the same in rats. That also took an hour and a half. And we could do it in marmosets. They wouldn't let me scan the marmoset for an hour and a half, so I got 20 minutes. There's a difference between these sequences. These first two are done in 3D. That means we're doing lots of phase encoding steps without size selection. Whereas the marmoset was done in 2D. And inadvertently, this had a completely different contrast mechanism to the first ones that I didn't know at the time. As a 2D sequence, it had a lot of slice selection, which led to magnetization transfer effects, which I'll just explain now, which gives us an alternative to the inversion preparation to get the contrast between grey and white matter. So here's a cartoon model of how magnetization transfer contrast comes about. If we consider the protons that we normally look at, it's the free water protons. So here's a picture of a load of protons just happily doing their thing. If I excite them and acquire my signal, they'd have a relatively long T2, T2 star, depending on what kind of sequence I'm using, and the signal decays relatively slowly. If we look at the frequency components that make up this signal, they're in quite a narrow band. Okay, so if I excite these with a particular frequency, assuming it's homogeneous, then that's a very tight set of frequencies, causing that resonance which decays relatively slowly. There's actually a second pool of water that we can't see. That signal decays very quickly. These come from protons that are tightly bound to the surface. Because they're bound to the surface, they can exchange energy very efficiently. They have a very fast T2, so fast that you don't actually see it. You can't see the signal from these protons. But in fact, although they're bound in layers to the surface, in equilibrium, these particles will exchange positions all of the time. So any particular proton may be bound or free, they're swapping exactly where they are. This very slow decay, this very fast decay, to decay so quickly, has a very broad band of frequencies in it. We can manipulate those spins by having an off-resonance RF pulse. So if I fire an RF pulse of quite a broad band of frequencies that's far away from the protons I normally see, those protons won't be affected at all because it's the wrong frequency to have anything to do with them. But the bound ones, they respond at that frequency, and I can saturate their signal so it wouldn't be seen. Of course, we can't see it anyway. But then when this exchange process that I mentioned happens, those saturated protons will not give any signal. So having fired the RF pulse, the signal from those becomes invisible. So when they swap with the free ones, that signal is going to be missing from what I detect. So if before I had the MT pulse, my signal looked like this, afterwards it would look like this, where there is a signal loss caused by the saturation transfer effect of my off-resonance pulse. The factors that affect this include the exchange rate of particles from the bound and the free pool, the intensity of the saturation that I apply, and the amount of those protons that exist. So if I put a beaker of water in the scanner, it wouldn't make any difference because none of those protons are really bound. If I put in something fatty with very many membrane layers, there would be a very large difference because there are many bound protons. And in fact, we will see a great difference between grey and white matter. So here's a section of marmoset brain, just with proton density weighting. There's no T1 here. If I apply the MT pulse off resonance, it looks like this afterwards. So the CSF, the white bits, for the physicists, isn't affected because that's all free protons. The white matter is affected a lot because there's very many bound protons. There's a great loss of signal. And the grey matter is affected to an intermediate amount. So this MT contrast will give me a strong differentiation between grey and white matter. So if we look at that, here's the proton density and empty image for a marmoset acquired in 3D shown together at the same time. This image, admittedly at 9.4 rather than 4.7 Tesla now, is acquired in about 7 minutes. So it's much faster, it's 3D, it's high resolution, and we don't get the rubbish signal that you'd get if you used an inversion preparation in MP-RAGE. So this is one method we can use to get high-quality 3D structural images from animals. And the point I want to make about this is, again, that when we think of translational imaging, often we want to do the same thing. So we take our human sequences and we put them in mice and we run the same thing because that's translational. Actually, that's a fallacious position because the scales are so different, the parameters are different. To make it translational, what you want to do is at every scale you have, acquire the best images you can, and that's to optimize for the contrast that you've got, optimize for the difference in magnetization properties that you've got. So much for structural imaging. What about fMRI? So one of the key problems about scanning mice, rodents, monkeys, as opposed to undergraduates, are that they tend to move. We need to fix them. So if they're awake, we'll have to use some kind of restraint, or they'll have to be very well trained not to move. Generally, everybody uses some kind of head restraint or head fixation for animals that are awake, which is possible. More commonly, in small animals in particular, anesthesia is required. Now, this has quite an effect on the fMRI signal, the particular anesthetic that's used. For example, isoflurane causes vasodilation, and it will interact with the bold signal quite a lot, and it depresses it quite a lot. The way that anesthetics work is obviously by preventing some of the connectivity in the brain, and some of the signals are just not there in the first place. They're blocked by the action of the anesthetic. The signal that you're looking at, effectively the plumbing in the brain, is highly dependent on physiology. So if you want to compare differences between animals in different states, you have to make sure that the depth of anesthesia is very well controlled. So things like the respiration rate, the heart rate. People doing this rather seriously would tend to use ventilation and look at a capnograph CO2 output to make sure that the depth of anesthesia is as close as possible in every case. Differences in blood flow will affect the bold signal that you get, and you could end up looking at differences between individuals that is just reflective of your anesthetic quality in terms of keeping it the same. In some cases, the contrast is increased by giving contrast agents such as superparamagnetic iron oxide nanoparticles. That dramatically increases the susceptibility difference between the blood and surrounding tissue, which increases the size of the bold signals. Another approach that can be used is multi-echo fMRI. And this is quite important, potentially, for small animal imaging because of the fact that susceptibility differences are much worse at high fields, and the brains are quite small, so almost all of it is affected by susceptibility issues. So, for example, if I show you 3 echoes acquired during an EPI sequence, so normally you would acquire slices through the brain successively. In a multi-echo sequence, you would acquire a slice and then again before going on to the next slice. And so you can get 3 different TEs, for example, at the same time. So the time difference between these would be of the order of 20 milliseconds or so, which is much, much faster than the changes you'd expect in the bold signal. If we look at the edge of the brain, so with the short echo time, here's about 14 milliseconds, you can see areas around the ear canal and at the front, the signal's already started to attenuate, but largely it's all still there. If we look at those same areas for the final echo, we can see those areas have completely gone. The TE that we want in different parts of the brain to maximize our sensitivity to the bold effect is to match T2 star of the difference we want to see. that's going to be different in different parts of the brain depending on what's going on. If we acquire these images, we can combine them with an adaptive weighting for every part of the brain to optimize the TE that we get, so we can get much better signal from before. Okay, so that can help you deal with these signal dropout issues. This is a picture of the optimal combination for these echoes here, which you can see for some parts of the brain looks just like the first echo, and for the central parts of the brain looks like the third echo. In fact, you can do something a bit better than that. If you look at the signal you'd expect in an fMRI experiment, so the signal that we see would be the stuff that's there, our echo time, and it depends on the T2 star. If we assume for a moment that the bold effect is entirely a T2 star effect, and we look at two different signals, so we compare rest versus activation. If we look at the difference between those, assuming that we had a change just in S0 or just in T2 star, here written as R2 star, it's the reciprocal, then looking at the change as a function of echo time, changes in the raw signal don't lead to any change in that ratio. Whereas real bold effect changes, if they are indeed carried in T2 star, we'd expect those to be amplified as a function of echo time. What we can do with that, if we take the time series from an independent component analysis or similar look at what the fMRI signal is, we can predict from the way the signal behaves whether that particular series is bold or non-bold. And so that's an approach that can be used. So it doesn't cost anything to do this approach, to acquire multi-echo fMRI, if your scanner's fast enough. Because it takes a little bit longer per slice, if your scanner can't handle the data rate, like mine, you have to have an upsetting TR. But for many people with good scanners, it's actually free to do this. So it's worthwhile acquiring the data. Moving to diffusion tensor imaging, again, you're going to have the same problems with motion. You may want to use physiological gating in your animal acquisitions. Because the white matter tracks are smaller, we're going to have harder diffusion. It's not going to be as easy for the water to diffuse within them. And so that means to get the contrast you need, you're going to need higher B values. But again, that's going to differ in different parts of the brain. So it's very useful in animals to use a multi-shell approach, which means you have multiple B values in your acquisition. So, for example, in the same acquisition to do 600 and 1500 or even higher. The EPI images, as I mentioned, because it's the high field and because the brains are small, have distortion. So here are some slices, just showing coronally from front to back of the marmoset, the sagittal and axial slices. If I blip my phase encoding gradients to acquire these images in one direction, they look like this. If I blip the other way, they look like this. So as I flick between them, depending on whether I start at the top of K-space and go down, or the bottom of K-space and go up, my image seems to change dramatically. This is because of the tiny bandwidth in the phase encoding direction. It's very sensitive to frequency differences. So we can correct for this if we acquire images in both directions, as I show you here. We can register those together to work out the displacement at each point to correct for this kind of distortion. So looking at various parts of the brain, just to point it out to you, you can see in this direction we have some stretching here, whereas here we don't. We've got some signal losses, so if you look around here, that signal is gone completely, not just distorted. It's because of the oxygen in the ear canal. You can't get the signal back, but you can correct for the shape. And so just an extra acquisition going up and down means you can fix these distortions and have images that which are much closer to the anatomy than before. So a software that does that is called TopUp, and it's included in FSL. But at the acquisition, you need to acquire those two different directions or a field map to do it. So, in conclusion, I just want to make the point that to translate MRI methods to animals is not always the case that you would want to run the same parameters. You definitely want to adapt them to optimize for the animal. Okay? For morphological imaging, the contrast mechanisms that you should exploit are probably going to be different to the ones you'd exploit in humans. For computational neuroanatomy, isotropic acquisition is really important, because that way you don't care so much about the particular angle of the head. It's easier to compare differences between individuals. Techniques such as multi-echo EPI can give you optimal T2 star contrast across the brain, and some extra calibration scans, which don't take very long, field maps, blip up-down EPI, they can help you relieve distortions. So that's it. Happy to take any questions you can have. Thank you.
Hi, thank you for your talk. I learned a lot today. So regarding diffusion, but instead of using diffusion as a way to study diffusion per se, I've been surprised of how much anatomy detail I can get with just diffusion. And you can get by with the tricks you showed at the end with like top up, or you can use diffusion encoding in a simple spin echo or something like that. So my question is, have you investigated the utility and efficiency of diffusion versus, say, MT or T2 or T1 for anatomical imaging?
The answer is yes for ex vivo imaging. When I've got brains in a pot, it tends to be a big pot with a lot of juice that's ruining my life because it wraps over into the brain. And a little bit of diffusion weighting, very small B value of, say, 50, is enough to kill what's in the pot and make the brain light up. But in terms of in vivo imaging, I haven't used it much as a contrast mechanism, but it would be very effective to attenuate CSF, for example.
Okay, thank you.
Okay, thanks.
Thank you for your talk. Is a macaque more like a large animal or a small animal?
It's more like a large animal for several reasons. Firstly, many of the people scanning macaques are using human scanners to do so, and you have a lot of intercalation between the grey and white matter. So one of the reasons why the T1 contrast is so annoying in rodents and such is just partial volume effects, and the differences are quite small. Whereas in the macaque, because the brain has a lot of white matter and quite big, juicy tracts, you don't have those partial volume effects to the same extent, and the contrast is still quite good. So I sort of deliberately didn't talk about macaques very much. They're in the middle, where you can get away with human sequences very well. Probably there's room to optimize them using some of the techniques I've talked about here to get even better contrast. But yeah, it's in the middle, I'd say.
All right, I have one more question. So what are your recommendations for kind of figuring out an anesthesia profile for bold imaging in animals? Is there any general advice you give?
If you don't have to anesthetize, don't. It very much attenuates the signal. What you want to have is a reliable stimulus where you can literally see the bold effect you've got and up your anesthesia so that you can see the extent to which the signal is lost. One of the problems is it's not uniform, and different brain circuits are affected to different amounts. For example, if you're looking at thalamic connectivity, most anesthetics work by shutting that down. So it depends exactly what you're looking at. I think it's still an open question, particularly looking at resting state signals where you don't have a strong stimulus. Some people say the signals are there and some people say they're not.
Well, great. I think with that,